Hello and welcome. Today, we're diving into one of the most important practicals you'll do for your AQA GCSE biology, investigating antimicrobials. You know, getting this one right isn't just about understanding microbiology, it's about absolutely smashing those tricky exam questions.
So here's the plan. We'll start with the core concepts, the big why. Then we'll walk through the all-important aseptic method. After that, we'll get into analyzing your results, cover those crucial safety tips and exam skills, and finish up with a super quick recap. Right, let's get into it.
Okay, first things first. Why are we even bothering with this? Well, this practical is all about figuring out how we can control the growth of microorganisms. And that's a massive deal in everything from medicine to keeping our food safe. So the huge question we're asking is this: what actually stops microbes in their tracks? We're going to be testing a few different things, and it is so important that you know the difference between them. First on our list are antiseptics. Now, these are the substances you'd use on living tissue. So think about hand sanitizer or what a nurse wipes on your arm before you get an injection. They're designed to be safe enough for our skin, but tough on germs. Next up, we have antibiotics, and here's the key difference. These are medicines that work inside your body. They're pretty amazing, actually. They're designed to target and destroy bacterial cells without harming our own. And finally, disinfectants. These are the heavy-duty chemicals you use for cleaning surfaces, like your kitchen worktop. They're way too harsh for your skin, but they are fantastic at killing bacteria in the environment. Right, onto the method itself. This section is all about technique. The word aseptic literally means without contamination, and trust me, it is absolutely critical. If you get this wrong, your results will be completely useless, and even worse, you could create a serious health risk. So, why all the fuss about being sterile? Well, it boils down to two massive reasons. First, you need a fair test. You don't want any random microbes floating in from the air messing up your results. But second, and honestly, this is the big one, safety. You do not want the bacteria you're trying to grow to escape and contaminate you or the lab. So here's how it's done, step by step. First, you take your inoculating loop and stick it in a roaring Bunsen flame until it glows red hot. That kills everything on it. Then, you use that loop to pick up some bacteria and transfer it to your agar plate. The goal is to spread it evenly all over the surface, creating what's called a lawn. After that, you'll place little paper discs, each soaked in a different substance, onto that lawn. Finally, you just tape the lid shut, label it, and stick it in an incubator. Now pay attention, because this is a massive safety point and a favorite for examiners. You must not seal the petri dish all the way around with tape. Just use a few small pieces. Why? Because this lets oxygen get in, and that stops some really nasty anaerobic bacteria from growing. Those are the ones that can thrive without oxygen and are often much more dangerous. Okay, fast forward a couple of days. You've incubated your plate. What now? Well, it's time to analyze your results and see what happened. We're looking for clear evidence of which substance was the best at stopping those bacteria from growing. The key thing you're going to measure is something called the zone of inhibition. It's that clear circle around one of the discs where no bacteria have managed to grow. It's basically a bacteria-free zone. And the rule is super simple. The bigger the clear zone, the more effective that antimicrobial is. Now, if you really want to impress and get those top marks, you've got to be precise. Don't just look at it and say, that one's bigger. No, you need to measure the diameter of the zone in millimeters. And even better, calculate the area using the formula pi r squared. This is a much more accurate and valid way to compare them. All you have to do is have the diameter to get the radius, square it, and then multiply by pi. Simple. And once you've got that data, you need to present it properly. A bar chart is absolutely perfect for this kind of thing. It gives you a really clear visual way to compare your results. I mean, just looking at this chart, you can see in a split second that antibiotic B was the clear winner in this experiment. Okay, so doing the practical is only half the story. To really secure those top grades, you've got to think like a scientist. You need to be able to evaluate the method, think about the risks, and spot any potential errors. This is where the marks are really won. Here is an absolute classic exam question. Why do we incubate the plates at 25 degrees Celsius in a school lab? Well, it's because 25 degrees is warm enough for bacteria to grow, but it's cool enough to discourage the growth of dangerous pathogens that just love human body temperature, which is 37 degrees. Trying to grow cultures at 37 degrees would create a serious biohazard. Let's quickly run through a few more essential points. Number 1, safety. Once that culture has grown, you never ever open the plate again. It gets sterilized and disposed of safely. Number 2, fair testing. You have to make sure every paper disc gets the exact same amount of the antimicrobial. And finally, you have to use a control, a disc soaked in sterile water. This proves that it's the actual chemical doing the killing and not just the piece of paper.
Right, we have covered a lot of ground here, so let's just take a moment to pull together the absolute must-know facts from this practical. Think of this as your final checklist for the exam. Okay, your key takeaways. 1. Aseptic technique is non-negotiable for safety and for getting results you can trust. 2. You incubate at 25 degrees Celsius for safety, never higher. 3. You measure the zone of inhibition, and if you want the best marks, you calculate the area. And 4. You always, always, always use a control. Nail those points and you'll be in a fantastic position. And just to leave you with something to think about, this practical is a model, right? But the real world is obviously a lot more complicated. So just because an antibiotic works brilliantly on an agar plate, what other things might change how well it works inside a person? You could think about dosage, side effects, or the huge problem of antibiotic resistance. It's a great question to help take your understanding to that next level.